今天很高兴能够邀请到何雅琪医师博士以及现在他是耶鲁大学的副教授来帮我们进行今天这个演讲。那我很简单的介绍一下何医师。对何医师他是成大医学系毕业，然后在台大担任内科的住院医师，主要是在感染科的这边。然后之后台大云林当主治医师好像是1年，然后这期间R4、R5也念了临床医学研究所，然后之后到美国的 Johns Hopkins University 念 PhD。那 PhD 的时候是在全球最 top 的 HIV 研究的实验室之一，那里念 PhD，然后之后PhD 完了之后 Postdoc， 然后Research Associate 的讲师助理教授是在 Hopkins 直接就一路做下来，然后之后到耶鲁，然后副教授助理教授、副教授这样子来做。他在每个阶段几乎都是最顶尖的，从医学生的奖项、住院医师的顶尖的奖项，然后 PhD 的奖项、Postdoc 的奖项、Young Investigator 等等的。然后各位也可以看到他的研究上得到很多 grant 的肯定，然后 paper 发在一堆很重要的 paper。那我想重点就是他研究的是重要的主题 HIV 这么重要、神秘又特别的 virus。那在这么竞争的主题里面，他又是做的人家已经做那么久了，但是他又有一些很新、很突破性的一些研究切入点。那今天譬如说今天跟大家分享的会是 single cell multi-omics 这个 approach 去解决 HIV 的一些重要问题。那我们很高兴今天有机会听到雅琪的演讲，那他也一定会非常乐意接受大家之后的提问，或者之后有一些合作的可能。那我们欢迎雅琪。
谢谢所长，谢谢大家来，就是this is really my great pleasure，就是我能够来这边，谈一下我们所做的一些东西，然后有很多我看到这些 audience 都是我们以前一起，比如说就像住院医师，或者一起念研究所，或者是比如说我们家的电视最开始是学长给我们的等等，就是我就是觉得，就是台湾有很多人就是对研究有兴趣，然后我们互相可以彼此学习，我觉得这是进步最快的方法。那不过我开始之后，我会得换成全英文，因为我已经不太知道这些就是学术上的 term，如果要换成中文、英文，你可能听起来也会很奇怪这样子，所以那我会试着我会用全英文讲，但是你如果有问题的话，用中文问，这个是没有问题的。那个附带一点是我大概上个礼拜刚接我们微生物所的博士班这个部分的所长，所以如果有一些学生有兴趣想要申请博士班或者是博后等等的话，可以跟我联络。有一些就是美国比较习惯的方式，对于怎么样筛选学生、怎么样找学生，有一些特殊的方式，我可以私下再跟就是学生或者是就是其他人谈这样子。So, my job today is to share with you how we approach this problem called HIV persistence using single cell multiomics. So my goal today is not to tell you everything about HIV. My goal today is try to sell you an approach so that hopefully you can take this approach to ask questions on the research field that you're interested in. Just to thank my lab first, that all of these results are coming out from my lab members. It's because of these young scientists driving all these fantastic research that allow me to present our work over here. So the first question that may be relevant to all of us is how do we explore into a new field, independent from my previous mentor, for example. For example, you've been to United States and you went to this famous lab, you come back home and try to do something, but how do you gain independence? So what I learned is that if you don't have ideas and approaches different from your mentor, it's not about, oh, this is mine, that is my mentor's. No, you need to come up with yours anyways. Otherwise, you're probably not competitive enough in the long run. And the second problem is that typically people use the single cell RNA-seq approaches because, all these papers have the single cell methods, and then I'm just going to do this because it just looks like a fancy and colorful thing. So I'll just do it too. The answer is no. You're better off just don't do it or just throw your money directly into the sink. Otherwise, you're going to have a lot of results, wasting money, but not knowing what you're actually doing. You're going to publish some Me Too study or mediocre studies or even studies that cannot be reproduced by anyone else. And the third question is, how can this fishing, explorative, correlative, single-cell RNA-seq-based profiling answer hypothesis-driven biological questions? So the key really is to ask the question first and see if single-cell methods can answer this question instead of just do whatever and then put single-cell in this. And when you have the question, you need to tailor your single-cell design and bioinformatic analysis to answer the questions. It cannot be you send the data to the core give you back beautiful plots, and you're trying to publish this. This is not going to work. Just are lessons we learned. So initially 2013, 2017, when I was at Johns Hopkins, my goal as a PhD student is I developed this method to sequence HIV in full length. And then 2017, I tried to sequence out, for example, splice HIV RNA. We use PacBio methods to learn about deep sequencing. I'm feeling that maybe this is just my career. I'm going to sequence the HIV reservoir all my life. But fortunately, I went to this meeting called HHMI meeting, 2013, that there are lots of great speakers giving their fancy research progress. And I heard two talks that changed my life. One from Avi Regev, the other from Howard Chang. Avi Regev just published this paper back then by Alex Shalak, who is a superstar, and we really learned a lot from him, saying that that was the beginning time that people can do single-cell profiling using, say, drop-seq, ink-drop, all these seq-well, fancy single-cell methods trying to understand biological processes at the single-cell resolution. So this is so attractive. And it was like, wow, single-cell, beautiful. So I asked my mentor, Bob, can we do single-cell on this? This is just beautiful. And then he said, yeah, Yachi, there's only 1 in a million CD4 T cells having HIV in there. Are you going to sequence 1 million cells just to get that one cell out? And you're going to sequence how about 50 million cells? $1 each? US dollars. That's not going to work, right? Huh, okay. Then the second thing I learned is from Howard Chang. So Howard Chang gave a talk. At that time, what I was talking about is about direct conversion from, for example, hepatic cells without going back to polyprotein stem cells. You could convert that directly into like neurons with action potential shown in, say, 13 days. It's because of certain epigenetic regulations. Well, that's just beautiful. I love this too. So these are two talks that changed my life. So the lesson I learned is that go to these good talks. It's not just about writing down things and memorize them as what typical clinicians or medical students would do. Just memorize. No. The key is not to memorize it. The key is to use their ideas and approach to solve the problem of your research, and plant those seeds in mind. And maybe another day they will be able to answer the questions you have. So then I went on and trying to answer this question called, how do we identify these HIV-infected cells, because they're really rare, like a 1 in a million cells. So back then when I was at Johns Hopkins as a postdoc or early faculty, what I did is I used HIV RNA as a surrogate, because HIV-infected cells don't have cellular markers, so easy. So if I activate these cells, they'll express HIV RNA, then I can design probes at 5' and 3' HIV, then we can see this in flow cytometry and sort this out for single-cell RNA-seq. So how do I develop this out of nowhere? The answer is I read papers. I read this paper by Sandy Clam, Nature Methods, saying that, there are ways that you can use single molecule fish to stain RNA and look at their transcriptome. But they have not reached single-cell level, not single-cell RNA-seq. I say, well, if you can use single molecule fish to stain cells, I can design HIV to do this. And if I make a few steps just to make sure that my reagents are all RNAs free, then maybe I can sequence out HIV. Yes, so this is what we did. Not only we get the single-cell transcriptome, but we also found HIV splicing into the host, that we can see that when HIV integrates into a cancer gene, that compared with this uninfected cells, which has nothing happening, HIV integration into the cancer gene upregulates the cancer gene expression downstream of HIV integration site, but not upstream, saying that during this HIV integration event, HIV dominates over the host promoter, and now it's HIV that drives cancer gene expression. So this is awesome and this is fun, but there's backstory about this. The backstory is that developing this assay from scratch is very hard. I remember this so clearly because this is July 4th, a national holiday in the United States. That this was the day that I sequenced out this cell that, I started doing these like washing experiments 9 a.m. till 6 is all done. So I sat through the sorter in BL3 from 6 p.m. to 6 a.m. all night to sort through these rare single cells. And I know this so clearly because I see these fireworks at in Harvard Johns Hopkins and feel like this looks like my fax blot actually. You see this red and blue thing. That looks nice. But the lesson I learned is that academic career is hard. It's like climbing a mountain, and we frequently get lost. We need a lot of resilience to go through all these. The reason why we're persisting is because we need to make sure first this is an important question. Not any question you can approach, but this is an important question. For example, solving HIV latent reservoir. We're ready for it, and we're ready to take challenges. And even if we fail, just like the whole night through, that lots of cells just didn't go through, the sort of clock so many times that I still enjoy the process, testing my questions. I just can't wait to see the result. And I'm not a robot of my advisor, and my students are not a robot of me, that we're just pursuing questions at very different level, pursuing truth. Then I started my lab 2017 at Yale University, and I happened to be so fortunate to have this talented grad student named Jack Calora. All right, he started PhD just the same year as I started my lab. All right, so that's great. He joined my lab initially trying to work on HIV and host chromatin interactions because HIV integrates into human genome, so he wants to understand what happened. So during the time he's reading papers, he said, I want to do chromatin conformation captures in 3C, 4C, and high C. These are just those like a genomics approaches. Then when we're reading papers, he said, "Oh yeah, actually, see, there's this Howard Chang paper saying that high chip is way better than high C." We said, "Yeah, I love Howard Chang. I heard his talk. Yeah, let's just pursue this method." So we did high chip. Great, but then we keep just following Howard Chang's paper, then found this guy named Ensu Setpathy. So he gave a job talk at Yale, and he talked about so many fun things at that time. He said, "You know what? You are doing single cell. I am doing single cell RNA-seq plus ATAC-seq in the same cell. Wow." And when you think this is fancy, he's doing something else. Not only just single cell RNA-seq, they can track T cell receptor sequences in addition to the transcriptome. Oh wow, these are just fabulous and fabulous, and that's the premise of our immunity paper. And the reason why we can pursue these approaches is because we have this grad student that his curiosity, autonomy, and the pursuit of scientific excellence and rigor just really drove me to just enjoy reading these papers, exploring into this. Again, as you can tell, this is totally different from my previous mentor Bob is doing. And I'm exploring with my student, and this is totally great. So then he said, actually, here's this paper that they can do single cell RNA-seq and surface protein together. I was like, yeah, that sounds fancy, but it sounds like enough money. I'm not going to do it. But then, see, November 2018, he found this bioarchive paper. See, like a half a year before it's fully published. He said, Yachi, more than that, they published new stuff. See, not only they can look at single cell RNA-seq, they can look at surface protein, and now they can track T cell receptor sequence all together. And it was like, wow. It's fancy. It's not just because this is fancy. It's because I have a burning question called HIV latent reservoir undergoing clonal expansion in my mind. And I know this is the method that can answer my question. So I say, we should do it. But how much does it cost? He said, let me figure out. So here are the protocols, totally doable. Your reagents cost, and we do this many samples, we set as control. Yes, we can do it. So we did it. 2018. Easy. So we use this new published bioarchive paper trying to understand C4 T cells from HIV-infected individuals. We get this beautiful rainbow plot. You see all these clusters, and we have all these beautiful heat maps. I presented these results at NIH Gates meeting in July, in which Alex Shalek is in the front row and Tony Fauci was sitting next to me. So I was so excited about our result because we are the first people characterizing C4 T cells from HIV-infected individuals. The answer is no. This is nothing to be proud of. You will see lots of papers having figures like this. Wow, rainbow plots. Wow, 27 clusters. Wow, heat maps. These means nothing. Nothing. Don't be satisfied with result like this. Basically, you have single cell data, you send it to the core, came back with this. You said, oh God, what did I learn? Because these fancy, beautiful papers has no biological insights in them. Having 27 clusters, and so what? I can divide into 57 clusters. I can have them into 7 clusters. It doesn't mean anything. Don't be satisfied with this. Many people starting the single cell field started with this, oh yeah, this is great. No, it's really bad. There's no biological insight like this. You need to have a bioinformatics approach to answer, to find out what's in this. So when we have this, and now we know what it is going on, if we see the single cell paper saying, oh, we find a signature of this, or we identify a novel cell type of that, this pretty much means that they don't have biologically significant findings, so that they just claim something similarly novel, but actually has nothing to it. So with that, we explore some 2-3 years into single cell based kind of these approaches with all different ways we're trying. I feel like this is a unique niche for my lab because I started looking at HIV sequencing, and then I moved on into clinical samples using a translational approach. But instead of HIV itself, I'm moving on toward the cellular transcriptome. And with that, I can try to understand HIV and host interactions. So these are all the talks I give, and some are what I'm giving. Like these Europe talks are what I'm going to September and November after the lockdown. So I'm able to use this approach to answer many different questions with many different collaborators because this is an approach, and we are going to establish this method that everyone inside or outside the field can easily reproduce, can easily use. And this kind of open spirit to share our method is what the single cell field, like Rahul Satija has been advocating, that we want to share, we want everyone to use this, and it should be all reproducible because we can all find biological findings together. And having interest alone is not enough, but fortunately NIH is very supportive on this. So these pink ones are the ones that serve as PI or MBI, basically principal investigators on the major programs I'm pursuing. My core focus of research is looking at the clonal expansion dynamics of HIV-infected cells, looking at HIV host interactions at the 3D chromatin level. But we need expertise from single cell bioinformaticians to help us approach these questions better. So we have single cell grants in the U1, UM1, and then we have an MPhAR grant with machine learning expert David Van Dyke. We recently got this PO1 to collaborate with Jonathan Lee, who works on post-treatment controllers, tri-specific BNAbs, and all the other integration site methods together. So in addition to these, we can apply these approaches to a basic science kind of approach to look at spatial transcriptomics with this structural biology collaboratory, or with more clinical collaboratories looking at HIV cure using CD8 T cells or BNAbs or CAR T cells, and other ongoing collaborations, showing that the single cell method is an approach, but we are not really the single cell people making those techniques ourselves, but using it to answer biological questions is very, very powerful. So I'm going to tell you this story that's published. This is, again, by this very talented grad student, Jack Laura, that this is his thesis project is his talk given to only one PhD student per year in this prestigious HIV meeting. This is on HIV host chromatin interactions, but the side project, which is this immunity paper, is posted as the back cover of this meeting's program book. He still has another CRISPR paper that was just accepted to JV that I'm doing proof right after this meeting. We need to submit this out, but with this. We can show you what are the questions we have, how do single cell approaches answer our questions, and by the end, I hope that this approach may be can be applied to the field that you're interested in. So here's my question. My question in my field is that HIV infection, if untreated, leads to AIDS. But fortunately, we have antiretroviral therapy. So all HIV infected individuals taking antiretroviral therapy can have undetectable viral load, can live a relatively uneventful life, and have a relatively normal life expectancy. But whenever this person stops antiretroviral therapy, even after 20, 30 years of antiretroviral therapy, the virus will come back, or viral rebound. So basically, by current guidelines, every single person having HIV needs to take antiretroviral therapy lifelong. And why is that? This is because HIV live in this HIV reservoir, HIV infected cells. They're super stable, have a long half-life. That if you take antiretroviral therapy 20 or 30 years, trying to wait for these latent reservoir to disappear, the answer is it's going to take 73 years. So, of course, everyone needs to take antiretroviral therapy lifelong. But but that's okay, right? That's okay. Say if I have diabetes, I just take one pill a day and keep following up. I can have a relatively normal life. So what's the problem with taking one pill of antiretroviral therapy once a day? What's the problem? The first thing is that people having HIV are still having chronic inflammation with increased cancer risk, increased cardiovascular diseases, and accelerated aging. And second is the cost of these antiretroviruses, given that how much it costs per person, plus having 38 million of people worldwide having antiretroviral therapy, this is an intimidating burden. And the most important thing is actually the stigma that HIV comes with. For example, if I tell you I have diabetes, you say, "Oh, you have diabetes." Then if I tell you I have HIV, people may just jump out. People may defriend me. People may not want to talk to me. People may think that, oh, I have some problems. So there is a stigma that we're still fighting against. Through our understanding of from basic science to translational research to clinical trials, the HIV field had moved forward, curing 3 or 4 patients, including the Drusilov patients, like till now for the past 30 years, using hematopoietic stem cell transplantation, using this special bone marrow blood stem cells called CCR5 delta 32. Basically, if you infuse people having this mutations mutations in co-receptor, then HIV wouldn't infect this new implant, and that's why people can be cured of HIV. But this is so hard. This is not feasible for everyone. So we need to find other ways for a cure. People have been working on this for so long. So why is it so hard to develop an effective HIV cure strategy? Here we go. Here's a CD4 T cell. HIV will infect CD4 T cells and integrate into the human genome as an integrated DNA provirus. Now, what happened is that, sorry, there are problems with this. So what happened is that we're trying to target these HIV infected cells, but unfortunately, once HIV is integrated into the human genome, it may enter a latency state that it does not express any viral protein so that the immune system cannot see this infected cell. So currently what the field is trying to do is to use the shock and kill strategy, using these drugs to reactivate HIV so they will start to express HIV viral proteins so that supposedly this infected cell can be recognized by cytotoxic CD8 T cells and be killed, or the cell would just die of viral cytopathic effects because these viral proteins are toxic. However, there are problems. So presumably HIV infected cells should die of viral cytopathic effects. But no, they don't die. Why? And second thing is that CD8 T cells, whenever they see an infected cell, they should be able to kill this very easily. But the answer is no, they don't always kill. What prevents CD8 T cell killing of the infected cell? And the third thing is that HIV infected cells don't just sit there and persist lifelong. They actually proliferate, from 1 into 2 into 4 into many, many. So not only they have a long half-life, but they proliferate. This is a process called clonal expansion, that from one cell into many, many cells. And this is the scariest part, that they're increasing over time. And how do we stop this process? So we need to stop this process, but the first step is that we need to understand how HIV persists in these infected cells so that we can specifically target these infected cells. However, we have this problem, the 1 in a million problem, that basically in people having HIV under antiretroviral therapy, only 1 in a million CD4 T cells have these replication competent HIV in them. and these cells do not have cellular markers that can distinguish them away from other cells. For example, if you give me a bulk of tumor cells, I can just grind them up and single-cell RNA-seq them, easy. But if you give me blood samples from HIV-infected individuals, only one out of a million is HIV-infected, having replication content virus, but the others are just uninfected. So you take these cells for bulk RNA-seq for anything you want to do. This is just looking at 99.999% of uninfected cells. It's not helping anything. And the second problem is that HIV residing CD4 T cells, but these CD4 T cells are not homogeneous. Basically, they are very heterogeneous, that they can be having Th1, Th2, or Th17 phenotypes. They can be naive or memory cells. They can be exhausted, can be activated, have different antigen specificity, or different proliferation capacity or cytokine responses. This is so complicated. So initially, we think that finding the one immunity for T cells in HIV-infected individuals is like the needle in the haystack problem. But later, we found that it's actually a needle in the needle stack problem because these cells don't have any markers. And with the heterogeneity of these CD4 T cells, we think that finding HIV RNA positive cells in the middle of the heterogeneous CD4 T cells is like high resolution understanding of the black hole in the middle of the galaxy. So this is very hard. And that's why we want to take the single cell approach. Instead of looking at bulk transcriptomics, just like smoothies, by looking at single cell RNA-seq, we can tally down all these individual cells to look into the heterogeneity of this population and identify the rare CD4 T cells. So the lesson here is that if you want to use single cell approaches, I want you to ask yourself that, why do I need this? Does my data set or the cells I'm interested in are heterogeneous? For example, brain cells, highly heterogeneous. Like embryonic development, highly heterogeneous, yes. Or am I looking for a rare cell? For example, the tumor in the middle of tumor microenvironment or the HIV cell I'm talking about. Otherwise, if you're setting in a homogeneous cell line for single cell RNA-seq, I would say you're just wasting your money. So our hypothesis is like this. We hypothesize that what drives the clonal expansion of CD4 T cells having HIV will be the same as the clonal expansion mechanism of the cells that the normal jobs that CD4 T cells do. So what do CD4 T cells normally do? Basically, here's a CD4 T cell. It has a T cell receptor. It's very unique to each CD4 T cell, like a barcode. Whenever there's an antigen being presented from antigen-specific cells, the CD4 T cell will proliferate. This is a typical antigen response. But HIV can infect antigen-specific cells and integrate into the genome. So if this antigen stimulation continues or recurs, then these antigen-specific CD4 T cells can proliferate, but same as these HIV-infected cells within these T cell clone. So we're going to talk about T cell clone a lot. So this is the same T cell clone having the same T cell receptor against the same antigen. But I am not saying that they're all HIV infected. I'm saying that a few of them, one of them, can be HIV infected, but this is a T cell clone responding to the same antigen stimulation. And we want to find out what the T cell clone is doing, because this is the driving force of the proliferation of HIV infected cells that we worry about. Easy. So the goal is to look at the immune programs that drive the proliferation of T cell clones having HIV in them. We can look at them in two ways, in vivo or ex vivo. In vivo means that you don't do anything in your test tube or petri dish. Basically, you simply collect blood samples during viremia and after viral suppression and profile them. And the ex vivo stimulation means that, okay, you can take the blood from the test tube, stimulate them to see which ones are antigen-specific cells and try to correlate with each other. So with that, we can find antigen specificity, T cell clone size, clonal expansion dynamics, and immune programs of these large expanding T cell clones that we're worrying about. So with that, we collaborate with Dr. Ann Dewar at Fred Hutch, having this beautiful clinical trial studies archiving blood samples during viremia. Basically, these are people at risk of HIV infection. They're being tested every month. Once they're tested positive, Ann would collect their blood samples and start antiretroviral therapy. And after 1 year of treatment that they have undetectable viral load for 6 months, they took blood samples again. So easy. If we take blood samples during viremia and after virus suppression and compare their immune profiles and T cell clonal expansion dynamics, we can see how HIV infection shapes the immune responses. And separately, by looking at antigen stimulation, the same blood sample, same tube, you can take them out and stimulate them to try to identify antigen-specific cells. And this method was developed by Shane Crotty, has been used extensively in his SARS papers, but it's very easy. Any antigen-specific cells may express activation markers, such as CD69, and T cell activation signal 2, such as CD40 ligand or CD154. By fact sorting, you can identify these antigen-specific cells and see what happened over there. So with that, this is by Jack, and starting from web bench study design, actual conducting the experiment to bioinformatic analysis using this paper he found in bioRxiv and do this. Easy. The simple rule of single-cell RNA-seq is like this. You need to put three things together into one bubble. Your cell, your barcode, reagents. That's it. And the reason why the 10x platform is so powerful is because the coupling efficiency is 80-90%, which is pretty good. If you use other platforms such as in-drop of dropsy, that's just 5%. Other, like a Microbial base, we bought like a few of those equipment. Some just don't work, like 5% or even less. So to see the reason why 10x is being so popular is because the efficiency is really good. Easy. You have a gem bubble, so you have your singles, your cell, and you have your bead to tell you the barcode and your reagents. Then you can characterize single-cell RNA-seq and to see your single-cell transcriptome. Easy. Everyone does this. The special thing about this method is that they can use typical FACS staining antibodies, just like those you look at, SFTC, PE, CD4, CD3, anything like those, to stain these cells. But instead of these fluorophores like FTC or PE, this comes with a DNA handle, a DNA barcode, so that you can see in this bubble what are these cells stained with to know their surface protein expression. Then we can map the transcriptome to HIV RNA to know which cell is HIV positive or not, and then we can map their T cell receptor sequence. So with that, we can look at single-cell RNA transcriptome. Then with that, you can define Th1, Th2, Th17, all these different polarization phenotypes. You can look at surface protein expression and define effector memory, central memory, or naive cells. So this is because some cells have to be defined by protein markers because CD45 RA and RO are defined by splice forms that cannot be captured by RNA-seq alone. So you'll need protein markers to define central versus effector memory. Then with that, we can map which cells are HIV RNA positive, and then we can look at T cell clone size. Easy. Basically, different CD4 T cells, say 100 of them having the same T cell receptor sequence, means that they come from the one same mother cell and proliferate into many. So this means that if you have many cells having the same single T cell receptor sequence, they're a clone. Then you can find big clones, say 100 cells, small clones, say 10 cells. So with that, you can color, I can see which cells are among the big clones, so you can color them as shown on this plot. So the first thing we can look at is the drivers of chronic immune activation during antiretroviral therapy to get single-cell RNA-seq. So this is a plot you don't typically see, and I encourage you to look at this because there are many new findings you can get without unlike what the core may give you. So instead of finding one gene, say PD1 is important or CDR4 is important, you can use de novo gene module identification by weighted correlation network analysis, WGCNA, to find genes that we can find TNF-driven gene expression like these genes persist despite antiretroviral therapy. And second, we can look at the signatures of HIV RNA-positive cells. For example, if you see, which cells are enriched of HIV RNA, the answer is it's very heterogeneous. It's all over the place. But if you look at, if we characterize a central memory or effector memory, we can tell you that in this inner circle, which is HIV RNA-positive cells, HIV RNA-positive cells are enriched in effector memory cells. Then if you look at differential gene expression, there are two ways to look at gene expression. One is called single genes. As you see in this volcano plot, you can see a lot of interesting genes like granzyme A or like a perforin. These are cytotoxic T cell genes. Or you can use gene set enrichment analysis, which means that you look at a defined gene, say those defined by January on exhaustion markers, a set of genes instead of one or two, a set of genes to look at your data set to see what are the gene pathways or specific area that's being upregulated. For example, we see that they're upregulating cytokine receptor interactions such as TNF and TGF-beta genes. Then we want to understand clone expansion dynamics of these T cell clones that we're interested in. Easy, you just color those T cell clones having HIV in there. Now you can see very clearly, if you see, do you see any patterns that these clones are enriched? Yeah, there are these two blobs. That if we just know what these two blobs are, this HIV containing T cell clones, then we are closer to cure, right? Hopefully. So first we see the antigen specificity because we can do stimulation and track all these cells back to the T cell receptor. We know that some are CMV specific cells, some are HIV specific cells. And then when we can next look at all the CMV and HIV specific cell phenotypes, then you can see that, wow, so CMV specific cells can polarize into this very functional granzyme BTH1s, while HIV specific cells are just all over the place, but they cannot polarize into a functional TH1. Then you can see, well, so what are the cells that are having big T cell clones? So these are the granzyme B TH1 cells that are naturally the immune effectors that are most clonal that we can see in these data set. So you can track at two different conditions, after stimulation and before stimulation. If you see there's after stimulation they're TH1, the answer is they're TH1 cells primed already in the unstimulated status. Then next we're going to track a few other things. So this is called T cell clone size. So clone size means big or small, like how many, big or small. 100 cells in a clone, big clone. 10 cells in a clone, small clone, for example. So we see that HIV-infected T cell clones are always larger in clone size during viremia or after viral suppression in unstimulated condition or in antigen-specific cells, which is memory cells, suggesting that somehow there's a mechanism that HIV RNA-positive T cell clones are just in bigger T cell clone size. Then the second thing we want to look at is called T cell expansion dynamics. Basically, 2 time points, and if they increase in number, we say they expand. If they're the same, we say they're stable. If they're gone or disappearing, we say it's contracting. All right. So by looking at two different time points, viremia to viral suppression, what happened to HIV RNA-positive T cell clones versus RNA-negative T cell clones? We find that they're extremely stable, peaking at 1, which means that their clone sites just don't change over time. The third thing we look at is, okay, you're telling me HIV-infected T cell clones are big in clone site. There are lots of them. They don't go away. They're stable over time. So what are these cells? What are the immune phenotypes? They're granzyme B TH1 cells. Again, because of single-cell RNA-seq, we can link all these things back together. So if you look at what are these genes that are differentially expressed in these big clones, these are all cytotoxic T cell genes, like granzyme B, granzyme H, granzyme A, perforin, NKG7. Wow, how does that happen? So we found that this is very interesting. We want to see, is that all coming back suggesting that it is, so HIV hides in these granzyme B Th infected memory cells. So Jack did this flow cytometry validation using blood samples from the SABIS cohort, which we did the single cell analysis on, plus a separate independent cohort that we recruited from Philadelphia that we just tried to see that if our hypothesis HIV enriches in these granzyme B infected memory cells, does it stay true? The answer is yes. In 2 different independent clinical cohorts, we found that granzyme B infected memory cells are enriched in HIV positive cells. Now, I gave this talk at one of the HIV meetings called CROI, and before my talk ended, then Brad, my collaborator, asked me, Yachi, did you check this factor called serpin-B9? What's this? And he said, you know what? These are cytotoxic CD4 T cells that if their job is that they are going to make cytotoxic granules to kill other infected cells, for example. But these granzyme B may flow back to the cell and hit the cell, so the cell have this intrinsic way to protect themselves from self-inflicted injury called serpin-B9. That's basically granzyme B inhibitor. So indeed, we see granzyme B positive cells having this upregulation of serpin-B9 expression, and this is what we think, that cytotoxic CD4 T cells secrete granzyme B. And these are cells that are naturally proliferative. They have really good antigen responses. Just like cytotoxic CD8 T cells, they use the same weapon to kill cancer cells, for example. However, if HIV infects these cytotoxic CD4 T cells, that if CD8 T cells want to kill them, HIV pretty much have the same weapon and the same shield protecting them from being killed by cytotoxic CD8 T cells. And with these ongoing antigen stimulation, they are going to trigger more proliferation of HIV-infected cells. So in summary, we found that HIV-infected cells are large in T cell clone size, are super stable over time, and they're residing in granzyme B positive Th1 effector memory cells because these cells are naturally more proliferative and they may resist T cell killing. So one last note that those are exciting and we're pursuing this direction, but people ask me, did you look at tissues? I said, single cell RNA-seq is great because you can look at all these single cells and tell you down what happened. But then if it's in a tissue, if you grind them up and sequence them, then you just lose the context of what cell is sitting next to what. So we're currently using Differential at 10 micrometer resolution to understand single cell transcriptomics at the spatial level. So 10x VCM is now at 55 micrometer resolution, which is not ideal, but maybe by the end of this year, about from August to December, they will launch a 5 to 7 micron resolution spatial transcriptomics. I encourage people who are interested to look into this. So this is what we did using Differential, a device developed at Yale, so we can put this device on a colon tissue and look at spatial transcriptome and identify which cell is which. So with that, I would like to thank our study participants, our collaborators, our funding agents, and mainly our lab because this is a very dynamic and very diverse lab, and having really good young scientists keep driving the progress of my research program. Advertisement, we have postdoc positions available now. Please email me. Thank you.
好非常谢谢雅琪很精彩的演讲。那这个当然只是他的研究里面的一部分。然后我想 single cell multi-omics approach 当你有想要回答的问题的时候，当它是适合你回答你的问题的时候，是非常的强大的。那不知道线上大家参与者有没有什么问题要问何医师和教授的？所以是凯健吗？杨凯健医师吗？对。
雅琪，
学长，
恭喜！对啊，这样 exciting results。其实我应该不应该是由我来发问，因为我其实不做 field。我在想，我看到就是这个雅琪演讲真的太精彩了，对啊。那我想请问一下，刚刚看到那一群就是在 HIV infected 之后，它会一直 persist， 然后 expand 比较快的这些 clones。那你怎么知道说他们其实因为这个 clone expansion 的速度可能跟它 proliferate 速度有关，也跟它被杀的速度有关系？所以你怎么证明就是说有没有 double evidence 证明说他们其实是 proliferating more or they got killed less？
We don't know， 我们目前不知道。就是如果你只是用 cell line model 可以解决很多问题 ，primary cell model 可以解决很多问题，但是那些跟真正在 in vivo 发生状况都不太一样，因为 in vivo CD8 T cell killing 在每个人都不一样，所以 it's a very good question that how do we know if they're proliferating more or they're just being killed less? So the answer is I think that both may happen。就是现在会觉得两件事情可能是同时发生的。那如果你刚好是you can proliferate more and you happen to be killed less over 10 years of course, I think those are like highly selected. Just both of them. 这事实上还没有人知道，而且 NIH 刚发了一个 RFA， 就是8月7号 deadline 嘛，要大家来研究这件事情。The answer is we don't know yet, and it's a good question.
那请问一下，它 granzyme B expression 增加，或者说你刚刚说它的那个这是 cytotoxic T， 那它怎么 defend against 这个就是会 recognize 这些 cell 的 CD8 T cell？ 因为看起来好像是他的意思是什么，要杀回去吗？还是说他怎么不被杀还是什么事？我不太了解这个当中 interaction。
呀，就是 cyto 它的 CD4 T cell 是这样子，比如说如果你有 TB infected cell， 或者是有一篇 paper 讲的是 bladder cancer， 就是 cancer cell TB infected cell， 或者是有一些就是 squamous cell whatever， 就是有一些cancer cell， 那基本上如果有一个 cancer cell， 那 CD4 cyto 的 CD4 T cell 会来 recognize 应该把它杀掉这样子。但是这是通常发生的事情。但是如果这颗 CD4 T cell 刚好是 HIV infected， 这时候应该有另外一颗 CD8 跑过来，就是虽然你要杀它，可是你如果是 HIV infected， 我应该把你吃掉这样子。就是 HIV infection 这件 件事情还是不能追，所以 CD8 T 小会应该要追这些 HIV-infected cell。但是它的问题是 HIV 自己本身有一些 mechanism 可以 evade CD8 T cell killing， 比如说他们有 NAF down-regulate MC presentation， 或者是他们有很多 escape mechanism 等等的，所以 HIV 是有一些本来 built-in 的方法可以躲得过，就是 immune evasion， 就是为什么 HIV 是没办法 kill 这样子。那但是你如果躲得特别好，那永远就追不上，等于是如果你刚好是在 granzyme B， 就是不只是 HIV 的关系，就是如果我刚好我是 granzyme B positive serpin positive cell 的话，你想要杀我，但是我自己我有 serpin， 就好像穿了一层保护膜包起来，那你就杀不死喽。所以我们是认为这些细胞它们比如说有很多层的方式让它们能够 evade killing。所以我们就会觉得说，那如果希望研究 cure 的话，是不是这些他们的这种 shield， 你知道，你有一个外星人要来攻击你，这个 shield 至少这个壳要把它打破，不然你要把它杀掉可能有困难，这样子。
了解，非常有趣，非常棒的 study， 恭喜。
还在做啦，不是很确定，就是这 serping 的部分是 transcriptome 的 data， 我们还不是很确定，我们还
要做function就对了，看 function 的一些
function 还没有做，得做。
了解。好，谢谢。
是。那还有没有其他的问题？我先问一个哈，就是雅琪，你说你用CDR3 来知道它是什么样子的 clones， 那在解析度上你需要同时看alpha chain跟 beta chain 吗？以及说只有 CDR3 够不够？你需不需要看其他 CDR1、CDR2 的部分？衍生下去的问题是，我不知道我是不是有漏掉一部分，就是任何一个 HIV/AIDS 的病人，他身上有大概会有多少种不同的 clones， 那这样 clones 的数目会影响他的 outcome 吗？或者是说他比如说你本来讲一个大概要73年他才会不见，但是这种 clone 的数目多的或少的，这有没有影响到它是不是更容易复发？对，
是，这是两个非常好的问题。那第一个问题是说，就是怎么样用 假设我们是要用 single cell 来 define 这个 T cell receptor sequence， 那这个是不是准确这样子？那有两个问题。第一件事情是，它的 region， 比如说 CDR3 这个是可以的，就是比如说它这个 package 刚出来的时候，它可以看到够长的一段的 CDR3 的 region， 所以可以。那你如果 alpha 加上 beta 是最好，比如说我们的 mapping rate 可以到大概90几 percent 这样子是可以的。但是有一些不同的 study， 比如说你如果用 single nuclear RNA-seq， 你大概只能 map 也许60到80%， 但是那些 data 还是蛮 useful 的，所以你只要能够 cover 到 CDR3 region， 基本上是大概100 base pair 附近，那这样是够的。有一些 benchmarking paper 去写说这样子是足够去区辨它的 T cell clone。那第二个问题是，如果只用 single cell RNA-seq 的数字读，其实是读不太到，因为你读，比如说你1万颗细胞，但是人的 T cell repertoire 出生的是大概10的14次方，那你真的去 people 大家做 paper 大概是至少10的6次方这样子，那只读10的4次方的这个 single cell 事实上是不够的，所以我们都会用就是它的另外一管血去做 bulk RNA-seq 去看它整个，比如说要看到1个 million 左右的 T cell receptor， 那我们才知道对过来这每一颗细胞到底是多少。那第二个问题是这些 clone size 到底是多大？哦，天哪，不晓得耶。这些 clone 是这样，就是 wax and wane， 就是有时候会随着 antigen response 会突然间 peak， 然后突然间 shrink， 就好像今天我如果得了 SARS， 我 CD4 specific cell SARS specific cell 会突然间变得很多，但是突然间变很少。那比如说像 Shane Crowley 在 SARS 的 paper 就有说，你的 naive cell repertoire 或者你的 clone size 会跟你的immune response 有关系，跟 outcome 有关系，或者是现在有一个 hot topic 叫做clonal hematopoiesis， 就是你知道就是在 pre-leukemia 之前的 clonal hematopoiesis， 那他们也都是用一些 single cell 的方法去看这样子。那 HIV 的，你知道我们就是6个病人，我们就拿去发 paper 了，所以 it's hard to say. We don't know yet. Maybe there is something. There's a clonal hematopoiesis paper in Nature Med about this, but that's still somewhat skeptical. So. 不晓得，非常好的问题，但是可以再多做一点，可能大家都还得再多做这样子。
是。好，那线上还有许多的老师跟同学，有没有其他的问题？
我看到冲志有一个问题了，是 OK， 他说 Congratulations， 还有没有人要来做博后的？2、3年吧，来吧。
我考虑一下。
有空来玩，对，不是啦，就是如果有需要啦，基本上是这样子。像有一个这个是谁有举手的？
对对对，那可以先自我介绍一下，然后提问题哈。
何老师您好，我是那个许方启，我现在是在 Johns Hopkins 当那个 visiting student。那我有一个 quick question for you， 就是 Thank you for your nice talk today。那就是在我们在做 single cell sequence 的时候，有一些东西，比如说像刚刚讲到的 coverage 或 mapping 的 rate， 那另外就是 SNPs 或是 RNA editing 的部分可能不容易看得到。那请问老师是，比如说我们如果用 long read sequence 来解决它 single cell 后端的 sequencing 的部分，你觉得这样的潜力是有没有这样的潜力？这样子？
非常有趣的一个问题。5月，这个问题是这样子，就 single cell RNA-seq 它通常是读的很短，比如说了不起300个 base pair， 然后 read depth 非常的 shallow， 比如说一颗细胞看25K 25000个 read 大概就差不多了，就已经没有钱了 basically， 所以这是一个好问题。那这个问题问的是如果你可以 sequence， 比如像 nanopore 或者是 PacBio 用比较 long read 的 sequence 你能 capture。事实上呢，so Cospin Harbor 我们5月多去参加的这个 meeting， 有一个人是用 nanopore 的方法，他还加了很多各式各样的方法呢，去看 RNA， 就是 HIV 的 RNA transcript， 然后再加上 cellular transcript。他有放在 bioarchive， 我可能晚一点寄给你，还是怎么样？可能还是没有完全，就是他有用几件事情。第一个是你要做 ribosomal RNA 的 depletion， 第二个是你的 RT 要够长，比如说他们用的是 marathon RT， 因为一般的 RT 像 super straightforward 会 fall off。那再加上你要有钱去 sequence 出来，这样子是可以的，否则是非常好的。比如说你如果有够长的 splice， 你可以看 splice variant， 你可以看 velocity much more accurate way。So， 就是没有钱啦。
Yeah, that's what I wonder for the splicing,
throughput 还没有
splicing
到。
side.
Yeah, throughput is not there yet. But
Okay.
I mean, if you have few cells, you can use smart-seq based methods and can sequence a whole lot. But if it's like millions of cells, just no money. It's $0.4 each cell for 10x. You know, 1 million cells is $400,000. Yeah.
这样子。
好，所以
US dollars。
所以目前还是 10x genomics， 您觉得就是最有效率，然后。
先说 full disclosure， 我没有拥有他们任何 stock 或者是什么 funding。No full disclosure。他的 developer 叫做 Howard Chang， 然后我说的这个 XIC 的这个 PI 叫做 Peter Smithberg， 从 neurogenome sample 现在跳到 10x 去了，所以 it's not just someone who's trying to sell you a thing to make money. They're really good scientists. So what can I say? They're really robust.
OK。那我另外想问的就是protein 的那个 level， 就是你有一些 antibody 可以知道 cell surface 的一些部分，那个部分当然是多了一个 dimension 的讯息。那它有办法，我不知道某一些 protein 其实在 RNA 的 level 你还是会看得到嘛？就是哪一些的 cell surface 的 protein 你一定得用到。因为多一个 protein 就多一部分的支出，或者是某一些其他的工具你就不能做。假设有其他的工具，它对 DNA、RNA 的 sequencing 很强大，但是它暂时还没有办法做 protein 的话。
几件事情，就是 protein 是这样子，一般人想要做 protein 的 single cell， 最开始就是做 Cytometry mass， 就是 mass cytometry Cytometry 这样子。Cytometry 给你大概40个差不多，因为金属的那个就是用光了，同位素就用光了。但是现在这个 Cyte-seq 或者是 ExCyte-seq， 现在你去 BioLegend， 它给你133个 antibody cocktail altogether plus your， 就是包括你的那个 isotype control 都在里面了。那它不再受到颜色，比如说 fluorophore 的限制，不再受到同位素的限制，你要多少你就多少，所以它只是卖一个 PBMC 的 cocktail 给你133给你做好了。当然你想要自己挑自己想要的100个50个，随便你挑，就是比如说 neuron， 不同的东西你想要挑你就自己挑，所以没有，事实上就像 FACS staining 一样，你拿细胞来就 stain and wash out， 所以因为 DNA barcode 是 unlimited， 所以你只要有钱，你要 sequence 什么都没有关系。那我在这里要跟大家 advocate 一件事情，就是有一个 paper 在 Nature Biotech， 叫 dogma-seq， dogma-seq， 这是 就是 Peter Smithberg 再加上 Aviv Group 他们两个合起来，就是发出来一个，就是他们现在可以看 protein， ATAC-seq RNA 三个一起看。那基本上我们是已经做完了，我们的博后已经在分析这个 data 这样子，但是不能跟其他做 HIV 的讲，谢谢你。就是我们已经做完了，然后我们真的就是 RNA-seq 加 ATAC-seq 我们已经做完了 ，protein RNA 加 ATAC 我们已经做完了，就是现在正在分析 data 这样子，是有这些 advancement 是可以 try 的。那就是可能还有更多可以 try 的，但是目前我只能跟你说 Excite-seq、Dogma-seq 这个是我们手里试过是没有问题的，非常的 streamline 可以做的东西。
是。好，那不知道各位老师同学还有没有其他的问题？
徐老师，许淑慧老师有举手。
好，那这是我们鸡蛋所的老师，
对，那个谢谢何老师的分享。这样，我的问题是说，当然可能真实的来自这些 HIV infection 的这些 sample 不见得那么多，不过因为一直都有跟 HIV infection 有关的这些 genetic 的 loci， 那我想问的是，这些人有没有看过他的 genetic 的 background， 他是很像的吗？还是说其实这个部分也不确定这样？对，这是我的问题。
之前是这样子，大概那是有个问题，大概2008附近吧。那时候我们刚进去的时候，有人这么做，他们去做了一个 GWAS study，找了数百人、数千人哦，就是找各式各样 HIV infected individual， 看有些人叫做 elite controller， 就他们得了，就是他们不会得病的，就是得了也不会发病这样子。那有一些人得了就会发病，那到底是什么样的 gene 呢？那答案找出来是有2个 gene 叫 HLA-B57 跟 B 就是 B27， 然后基本上就是你如果有这样的 HLA， 你的 CD8 T cell 会比较容易保护你的这些 infected cell 这样子。那当然你如果 CCR5 delta 32就是 CCR5 mutation， 你也可以比较不会得感染。但是说实在的，他们那个 GWAS study 发出来的result是大家都已经知道10年的事情，就是等于他们费尽心力做完了 GWAS， 得到是教科书上已经写的答案，所以答案就是oh well， 就是well that's probably it， 就是either 就是还是太 heterogeneous， 或者是that's just about it， 就是基本上 HIV 是一个非常容易 mutate 的 virus， 所以我们所知的可能就是这么多了，那剩下的可能就有太多的 individual diversity 没有办法用 GWAS 去抓出来这样子。
了解，谢谢。
是，谢谢。那还有没有其他的问题或 comment？
好像还有举手，然后你要直接点吗
？OK， 还是。
喂，
是。李美叶老师，对。
陈老师您好，
中研院的大咖，对，这核心实验室的，对。
谢谢老师精彩的演讲，真的是收获很多。不过我本身我不是 biomedical background， 所以我想用一个比较 side track 的方式来问。好，因为我们有在 operate NGS， 然后我们自己有在做 single cell， 可是常常有人来问我们，就是说他假如说单纯一个 surveillance 的一个这样子的一个愿景或者一个 goal， 他观察到了一个很有趣的现象，那 necessary biomedical。好，可是他假如可以 isolate 到 single cell， 那只是想 survey， 你看到说现象里面，假如说他有看到不同群，比如说从 histology 还是说从 physiology 现象，他预期或不同群，那可是那然后我们就讲说你必须有一些 marker， 就是讲很直白，你必须有 marker 帮你 identify 你分群之后的东西的意义。可是他说，可是我就是要找这些不同的东西。那有时候很难去 communicate 这一块，因为大家的领域不一样，好，我也不能 represent everybody， 所以想听听老师您的意见是说，当您碰到这样子的 inquiry 的时候，您会给什么样的建议 ？To what extent 一个 surveillance 的 project 在 single cell 用 single cell 的技术上面这种投资是可以有比较预期的效益？谢谢。
非常好的问题，我们最开始也是这样子。我只能说，在 biomedical 这一边的这个 investigator 这个 PI， 他必须先找到一个懂 single cell 的人问清楚，但不然的话下去都不知道在干嘛。首先第一件事情是，其实我非常同意，就是用 single cell approach 去找一些，就是做一种 hypothesis 的 search 这样子。比如说，你就老鼠有没有 phenotype 去做什么，也不太知道，就是一个 search。但是呢，会牵涉到你要多少 sample size， 你要多少 replicate， 那你的 sample 是不是 heterogeneous？ 所以第一件事情是他得找到会做 single cell 的人， 来问。比如说我们刚开始做的时候，我问的是谁？我不懂，我问的叫做 Alex Shalak。Alex Shalak 就是当年 Aviv Regev 手下第一个做出 single cell 的就是他。他在 MIT， 我带着 student 带着 Jack 跟 Christine 去他的实验室 present 我们的 data， 非常非常的 stupid 就 slate 的 data， 他一步一步告诉我们你应该要做哪些东西，必须要这么的 detail， 在非常早的时候第一年我们就做了。那第二件事情比较快速可以回答的事情是，这个很痛苦，就是我们比如说，假设我给你看说，我有27个 cluster，12个 cluster，those don't make sense。这个 biology 的这个 PI 必须要看。假设我今天说我 CD4 T cell 有 activation 跟没有 activation， 你的分析给我的时候，我必须要知道我的 activated cell 有没有 express 我想要的 marker。这种 sanity check 是 biologist 必须要做的。但是当 biologist 跟 bioinformatics disconnect 或者是讲不太清楚的时候呢 ，biologist 会觉得哦，你给我 data， 你给我100个 gene， 我就想去 validate。The answer is not gonna work， 就是你必须要先 go through 你的 sanity check。你要的 cluster TH1 有没有看到 interferon gamma， 这样对吧？它有没有 express T-bed？ 那这种这么 technical biological 的事情是这个 PI 要做的。那 bioinformatically 有一些方法可以试着去找出比较 rigorous 的方法。那有好几篇 benchmarking 的 paper， 我们自己 lab 在 discuss 的，比如说你的 batch effect correction， 我们试所有7种的 batch effect correction， 因为有一些可能会给你 false positive。第二个是你要多少个 replicate。那第三个是你的 differential gene expression 有非常多的 package， 哪一个比如说现在都在做，比如说 Wilcox T test 就已经很好了，比最开始的 Shura 的 D-Seq2 那些好非常多。就是那你要怎么样去 sanity check 说我的哪一个 pipeline 可以给你最好的 data， 这是 bioinformatics 可以说我试这6个 pipeline， 那你是 biologist， 你得告诉我你要的那个 positive control 的 population 这个 TH1 是否 expressed interferon gamma 跟 T-bed。那这2件事情交叉过几次才会 work， 那就是为什么我们的实验室自己有我们的 bioinformatician， 因为我们自己要让 R， 不然我们根本不知道跑出来的东西是什么这样子。快速的就是他们得去找真的做 single cell 的人谈过，然后试着跟你沟通，然后他们得试着找出来他们的 sanity check 那个 population 是什么，那个 cluster 是对的，那才能确定说我们找的 gene 是适合去往下做 downstream validation。
好，谢谢你。
好，那我想我们从雅琪的演讲知道的就是biomedical scientist 我们自己要知道要问什么问题，那这个问题是不是 single cell 的 data 可以来回答的，这个我想是最重要的第一步。然后要去bioarchive 或者是一些 meeting 去找到还没有 publish 的新的方法，或者都会晚个1-2年。然后第三个我觉得 connection 是重要啦，就是雅琪今天能够来跟我们演讲，那我相信未来有什么问题，有什么需要合作的，我就觉得都有机会。对，那还有没有其他的问题？
好像看还有一个人举手，你要不要点一下？
好，
还是有没有人要直接按 mute？
来，我们最后一个问题哈，来，请自我介绍一下，然后提问。
那个陈教授何医师好，那我是阳明交通大学柯泰明，然后那谢谢你的演讲，今天非常精彩。那我这有就是有带3个快速的问题，第一个就是那个for bulk RNA-seq 和single cell RNA-seq 的这样的一个串联，我觉得这是一个非常好的一个 strategy， 因为bulk 的 T cell receptor 其实是可以比较 comprehensive， 但有一个问题是说，在因为 single cell 它的量还是有限的，就是在你们在 match 的时候，以你们经验来说，其实是它可以 map 就是对应的很好吗？还是说有些时候其实就是 single cell 它所找到的不见得可以完全反映在 bulk 的 RNA-seq 的这个 data 上面，这是第一个问题。
第一个是 bulk 还是比较 rigorous， 因为 bulk 你可以看到1个 million， 那 single cell 你放进去多少它就是多少，比如说 10,000 for example， 那其实 10x 的mapping rate 可以到 90%， 那有其他的，比如说你如果做过就是有 fix 过的 sample， 比如说像就是 sequel 那附近出来这个叫 track-seq TREK track-seq Alex Shalak 出来的，用这种 这其他方式去硬去弄出来，可能到60%到80%的 mapping rate 大概就差不多了。那看你的问题，就是你60到80%，你有很多细胞，你还是可以做某一种分析。但是你如果只是想要看这个病人的哪一个 clonal type 多，就 bulk 就可以解决了。但是你要问说这个 clonal type 它是什么样的 T cell phenotype，那你就得进 RNA single cell 你才能够看到它的 phenotype。
理解，谢谢。那第二个问题是我其实刚刚也跟凯健有类似的问题，就是在 T cell clone expansion 的那一群是不是就代表有function？所以这个方式当然我们可以用不同的 clone type 跟基因的连接有一个关联性，但是是不是有可能是因为我们都已经知道它配对的 T cell alpha chain 跟 beta chain，是不是有可能把它clone出来，然后再去做一些功能性验证，有可能吗？是。
Here we go。我大概3月5号的时候要给一个演讲，3月3号的时候 ，Michelle Nielsen、Howard Hughes Investigator， 然后 Rockefeller 的一个大头这样子。他在3月3号的时候放了一篇 paper 在 bioarchive。那他们做的是这样子的，他们就是去看 HIV positive individual， 他们有几个病人，他们完全知道 T cell clone type， 他们就是用 T cell beta 去把它抓出来，然后抓出来以后就找到一大个 clone， 然后用那个 clone 去做 single cell RNA seq。所以有这个方法的，就是你如果知道你的 clone 特别是要什么，你可以把它用 fax 或者是用 bead 把它 select 出来。他们也是发现 HIV enrichment in cytotoxic CD4 T cells， 就是3月的时候跟我们发现一模一样的东西。那他们那个办法是有办法做，就是 downstream 的 analysis， 但是我们并没有刚好有办法可以 sort 这样子，所以那个方法是有的，那个是 Michelle Nielsen 的 paper 在 bioarchive。那我们两个可能都会往下做吧，我也不晓得。
谢谢。
不是很容易啦，就是要找到 specific 那个clone，有方法可以 select， 但是不是很容易。
那最后一个小问题是就是 for 你们找到的这些序列，就是比如说 for example 在 beta chain 的部分的这些 CDR 会不会跟之前在过往的别人用 bulk 的就是方法有找到 identical 或者相似的序列？
很有趣，就是我们刚开始拿到的时候，我们也很想看，就是比如说我们就去那个就是已经 publish， 像主要是 Mark Davis 他们有很多的，就是主要是这样 CDA， 就是 MHC1 publish 比较多 ，MHC2 publish 不是很多，因为我们做的是 CD4 T cell 这样子，所以我们有去试试看，去说这样可不可以map 到什么 existing 的，但是这实在太 diverse， 所以我们没有真的 map 到别人有 report 过的，但是 again 这些 participant 每一个的就是 genotype 都不太一样，所以我们可能说 OK， 那我们把我们的 deposit 上去，那有一些人会用一些 algorithm 去做某种 prediction， 但是我们是有 select 过 T cell receptor 是 antigen specific， 所以我们就放上去，那也许你可以用这个来做 validation， 或者是以后别人也许可以找到类似的东西。
很好，谢谢何医师，谢谢陈教授，谢谢。
好，那非常谢谢雅琪的精彩的演讲。